गुड मॉर्निंग स्टुडंट. नवयुवक शिक्षण प्रसारक मंडळ चापोली संजीवनी महाविद्यालय चापोली वेलकम you in e-learning platform prepared by myself Dr. Shubhangi Chavan, Assistant Professor and Head, Department of Botany, स्वामी रामानंद तीर्थ मराठवाडा University Nanded. Semester Pattern Curriculum under Choice Based Credit System CBCS for undergraduate program. Faculty of Science and Technology. Subject Botany, Year BSc First Year. Paper First, Viruses, Bacteria, Algae, Fungi, Lichens and Mycorrhiza. Now start to chapter. Continue. Bacteria. Last period to see ultrastructure of bacteria. Now start to reproduction of bacteria. Bacteria reproduce asexually and sexually. Asexual reproduction of bacteria. Asexual reproduction takes place by the binary fusion and by the formation of endospores. Now, by the binary fusion, fusion or cell division is the most common method of asexual reproduction in bacteria. The fusion takes place during the period of favorable condition. The process of splitting of bacterial cell into two new equal daughter cells is called as the binary fusion. The binary fusion is common role in bacteria. During the process of binary fusion, the bacterial cell elongates first, then the protoplasm divides mitotically and result into the formation of two parts. Meanwhile, the plasma membrane just at the middle of the cell and between the two parts of the protoplasm forms a constriction. The construction grows inwardly and finally forms a transverse plate between the two parts of the protoplasm. Now next, the plasma membrane of the transverse plate separates the two parts of the protoplasm. As the process processed, the cell wall form construction just at the middle in between the plasma membrane of the transverse plate. The construction of the cell wall grows inwardly and form a transverse wall between the two plasma membranes of the plate. The transverse wall thus formed splits the parent bacterial cell into two equal daughter cells. These newly formed equal daughter cells grow in size the bear at the new bacterial cells. Now, the fusion of bacterial cells occur as often a very 80 to 20 minutes under the favorable condition. A bacterial cell dividing at the rapid rate of 20 minutes will produce the progeny of about 47 into 10 into 20 cube in individuals in day at it worldly weight about 2000 tons. Such a fantastic rate of multiplication is because of the space limitations computations of oxygen in case of aerobic bacteria, adjacency and food supply, etc. Now the diagrammatic representation of binary fusion. Present diagram show asexual reproduction of bacteria by binary fusion. we can observe how nuclear material, that is DNA, divides into two cells that are equal daughter cells. Now, by the formation of endospore, the asexual reproduction by the formation of endospores is very common in the rod-shaped bacteria. The endospores are small, rounded or spherical, thick-walled unicellular structure. They are formed during the period of adverse condition. They are produced singly during the process of formation of endospores. The nuclear material of the parent bacterial cell become filamentous first. A ring-like investigation of the plasma membrane is just near one of the polar end of the parent cell. The investigation grows inwardly and finally result into the formation of a transverse spectrum between the small polar portion and rest of the portion of the parent cell. The spectrum separated the small polar portion from the parent cell. Now, the diagrammatic representation of endospores formation. This diagram describes step by step asexual reproduction by the formation of endospore. In figure A, we see vegetative cell with plasma membrane. In figure B, nuclear material start to divide. In figure C, initiation of transverse wall striated. We can observe ring-like investigation. In figure D, complete transverse wall appears in figure E, new wall formed around the endospore. In figure F, G and H, formation of endospores are completed. Now, a small amount of nuclear material also trapped within the polar portion. This polar portion grows in size, become rounded or spherical or cylindrical, thick-walled and behave as the endospores. When the endospores mature, liberated outside by the wrapping wall of the parent cell. Endospores is resistant and dormant and undergoes rest during unfavorable condition. After the return of favorable condition, it germinates and gives rise to new bacterial cell. Thank you, student.